You are standing right in the middle of a valley.
Right.
And it's the dawn of the 20th century. If you just look around, the landscape is absolutely breathtaking.
Yeah, it's gorgeous.
To your east, you've got the Sapphire Mountains, which are, rolling and relatively dry. But then to your west, you have the Bitterroot Mountains. And these are jagged, they're heavily timbered, they are cut with these incredibly deep, shadowed canyons.
Very different terrain.
Exactly. And running right down the middle of this entire valley is the Bitterroot River. So imagine you are a settler here in Montana, you brought your family out, you are clearing land, you're planting these big apple orchards, but there is a very literal geographical dividing line dictating whether your family lives or dies.
And that line is the river.
It's the river. If your farm is on the east side of the Bitterroot River, your life is, it's hard. You're a frontier farmer, obviously, but it's normal.
You're dealing with normal frontier problems.
Yeah. Right. Weather, crops, that sort of thing. But if you live on the west side of the river, nestled up against those steep canyons, you are essentially playing Russian roulette every single time you walk into the brush.
It's just an incredibly stark geographic demarcation. We were looking at a mortality rate of 80 to 90% for a very specific illness on that western side.
Which is just a mind-boggling number.
It is a staggering number. If 10 people contract this mysterious fever, maybe 1 or 2 survive. And for the longest time, literally nobody knew why it was happening.
So today's deep dive is taking us into a dossier covering a highly unusual, just fascinating chapter in the history of the U.S. Public Health Service, or the PHS. We are looking at the horrific Rocky Mountain spotted fever outbreaks in Montana's Bitterroot Valley in the early 1900s.
Yeah, it's a wild story.
It really is. Our mission today is to explore the actual science used to stop this killer. And we're going to examine a very specific historical shift. We want to look at how a makeshift laboratory set up in an abandoned schoolhouse and run by men who were taking just unimaginable personal risks eventually gave birth to one of the most sophisticated modern medical institutions we have today.
Because if you look at the overarching history of the U.S. Public Health Service, their role historically was predominantly administrative, regulatory.
Like enforcing rules and stuff.
Exactly. They were the ones enforcing maritime quarantines or conducting the medical inspections of immigrants at Ellis Island. They set broad sanitary policies. But the actual bench science, the microscopic discoveries, the isolation of new pathogens that usually happened in big European academic institutions or private laboratories.
Right, not out in the woods.
No, definitely not. The Bitterroot Valley represents a fundamental break in that pattern. Here, the PHS officers didn't just coordinate a response from an office in Washington. They went out into the frontier, they built the lab themselves under just abysmal conditions, and they did the foundational, highly lethal bench science.
Okay, so let's get into the realities of that valley first.
Yeah.
Because I feel like to understand the science, you really have to understand the terror that was driving it.
You do.
You mentioned the 80 to 90% mortality rate. But what actually happens to a person who catches this? The dossier refers to it as a mysterious illness, but what is the physical, the bodily reality for a settler in 1902 who comes down with this fever?
It is a brutal way to die. Rocky Mountain spotted fever, as we understand it now, is fundamentally a disease of the blood vessels.
Okay.
The pathogen specifically targets the endothelial cells, and that is the single layer of cells that lines the absolute interior of your blood vessels.
Oh, wow. So it's not attacking your lungs like pneumonia or, your intestines like cholera. It's attacking the plumbing itself.
That's a great way to put it, yeah. It attacks the plumbing. The pathogen invades those endothelial cells, and it just begins to multiply inside them. And eventually the cells become so packed with the pathogen that they literally rupture and die.
Oh, God.
Yeah. And when the cells lining your blood vessels die, the vessels begin to leak.
Which I'm guessing explains the spotted part of the name, Rocky Mountain spotted fever.
Yes, exactly. The characteristic petechial rash that appears on the patients, it usually starts on the wrists and ankles, and then kind of spreads inward. That is not just a skin irritation like poison ivy.
It's blood.
It's blood. Those spots are microscopic hemorrhages. It is blood leaking out of the damaged vessels directly into the surrounding tissue.
That is horrifying.
And as this vascular leakage progresses systemically throughout the body, the patient loses actual blood volume. This leads to profound hypovolemia, meaning there just isn't enough fluid for the heart to pump anymore.
So your blood pressure just bottoms out.
It plummets. And then the major organs, the kidneys, the liver, the brain, they are completely deprived of oxygen. This leads to multi-organ failure, shock, and eventually death.
Mm-hmm.
It is a rapid, incredibly painful, and deeply traumatic progression for the family to have to witness.
And if I'm a settler out there, the thing driving me completely out of my mind is the geography of it. Like we said earlier, if I live on the west side, my neighbors are literally bleeding out internally from this fever, but I can look across the river to the east side, maybe just a few miles away, and the people over there are generally fine.
Right.
I really want to dig into that puzzle. Why the west side? I mean, the river itself obviously isn't some magical, invisible barrier.
No, it all comes down to microecology. The two sides of the Bitterroot Valley just have very different topographies and microclimates.
Okay, how so?
Well, the west side, which is nestled against the Bitterroot Mountains, is characterized by these steep, heavily wooded canyons. There's a lot of moisture, very dense brush. And this environment is the absolute perfect habitat for a specific organism. It's called Dermacenter andersoni.
The Rocky Mountain wood tick.
Exactly, the wood tick. But more importantly, it's the perfect habitat for the small mammals that these ticks feed on during their early life stages. So we're talking ground squirrels, chipmunks, voles.
Got it. And the east side?
The east side of the valley, over near the Sapphire Mountains, is much drier. It's more open. It's relatively brush-free, which is far less hospitable to that specific tick ecosystem. So the ticks were heavily concentrated in those western canyons. And the settlers, they were essentially clearing land and building cabins right in the middle of a highly concentrated infected tick habitat.
It's like building a house inside a hornet's nest and wondering why you're getting stung.
That is exactly what it was.
So this geographic anomaly, this localized nightmare, is what finally forces the federal government to get involved. In 1902, the Hygienic Laboratory, which was actually the precursor to the National Institutes of Health, but at the time it was operating under the PHS, they sent an investigator named Dr. Julius Cobb out to Montana.
Did you? Yeah.
But Cobb is mostly doing, like, observational epidemiology, right? He's mapping cases, he's looking at the environment. He isn't the one who finds the actual killer. That happens a bit later, around 1906.
That's right. Cobb laid some important groundwork, but the real breakthrough comes with the arrival of a pathologist from the University of Chicago, a man named Dr. Howard Taylor Ricketts.
Okay, Ricketts.
Yeah. Ricketts is the one who transitions the investigation from just observational epidemiology to hard, hands-on laboratory science.
I want to break down exactly what Ricketts achieved here because he essentially cracks the two hardest parts of the case back to back. First, he proves the delivery mechanism.
Right. Through an incredibly rigorous series of experiments, mostly involving guinea pigs and monkeys, Ricketts proves definitively that the disease is not in the water, it's not in the air, and it's not contagious from person to person.
So you can't catch it from your spouse coughing on you?
Exactly. He proves it is transmitted specifically by the bite of the wood tick. He actually took ticks from the west side of the valley, allowed them to feed on healthy lab animals, and then watch the animals develop that characteristic fever.
That's pretty definitive. And then he goes a step further, right? He doesn't just point to the tick as the vehicle. He finds the actual passenger inside the tick.
Yes, he does. Using microscopy techniques that were honestly quite advanced for that era, he examines the blood of infected patients, and he examines the tissues of the infected ticks. And he identifies a microscopic organism.
What did it look like?
It was a tiny, rod-shaped bacterium. And this organism was so biologically unique that an entirely new genus was eventually named in his honor. They called it Rickettsia.
Rickettsia. Okay, let's stop here for a second because this is the part of the timeline in the dossier that always trips me up.
Sure.
Ricketts identifies the vector, the tick, and he identifies the pathogen Rickettsia. And this is all happening around 1906.
Correct.
But if you look at the history, a viable vaccine doesn't actually arrive until the mid-1920s. If you know the bug causing it and you know exactly how it gets into the human body, why does it take nearly 2 decades to figure out how to stop it?
That is the million-dollar question.
Yeah.
And it's because identifying a pathogen and actually manipulating it are two entirely different universes of biological complexity.
What do you mean?
Well, it's the difference between taking a photograph of a wild animal in the woods and successfully capturing and domesticating it.
Okay.
At the time, bacteriology had a very standard playbook.
Okay.
If you found a bacteria causing a disease, let's say the bacteria that cause anthrax or cholera, you could usually extract it, put it in a glass petri dish filled with nutrient agar, put it in an incubator, and the bacteria would just grow and multiply on its own.
Like mold on bread.
Exactly like that. And once you can grow it in a dish, you have a limitless supply of the pathogen. You can experiment on it, you can test chemicals against it, and you can try to turn it into a vaccine safely in vitro.
But I'm guessing Rickettsia wouldn't grow in the dish.
It absolutely refused to. And the reason why is something that stumped researchers for years. Rickettsia is what we call an obligate intracellular parasite.
Okay, break that down for us. Obligate intracellular parasite, meaning it has to be inside a living cell to survive?
Precisely. Unlike standard bacteria, which are free-living and can easily scavenge nutrients from an agar plate, Rickettsia has evolved a highly reduced genome.
Meaning it's missing some genetic code.
Yeah, it literally lacks the biological machinery to produce sufficient amounts of its own ATP, which is the energy currency of the cell, or to synthesize certain essential amino acids. It has to steal those resources directly from a living host cell.
Oh, wow.
So if you put Rickettsia in a nutrient broth or a petri dish, it just sits there and dies. It needs living tissue to replicate.
So the science of 1906 was essentially stuck. They didn't have the cell culture techniques we have today where we can just grow sheets of living human or animal cells in a lab to host a pathogen.
No, not at all.
To grow Rickettsia back then, you had to grow it inside a living animal or inside the tick itself.
And that made research agonizingly slow. It was incredibly expensive and profoundly dangerous. You are constantly handling live animals and live ticks that are infected with an 80% lethal pathogen just to keep your microscopic subject alive.
It's like you can only study the bomb while it's actively ticking.
That is a very accurate comparison.
And speaking of that danger, we really have to mention the tragic irony of Dr. Ricketts himself here.
Yeah.
Because he makes this monumental discovery. He literally gets the genus named after him, but he doesn't get to see this story through.
No, he doesn't. Ricketts was really emblematic of this entire era of micropunchers. They were working in incredibly close proximity to lethal pathogens with virtually none of the biosafety equipment we consider mandatory today.
No hazmat suits.
Barely even gloves in some cases. But it wasn't the Bitterroot Valley that claimed him, surprisingly. In 1910, his expertise was requested down in Mexico City, where there was a massive outbreak of typhus.
Which is related to spotted fever, but it's not the exact same thing, right?
Right. Typhus is caused by a different species within the Rickettsia genus. It's called Rickettsia prowazekii, and it is transmitted by the human body louse, not a tick.
Okay, got it.
So Ricketts went down to Mexico to study the pathogen. He contracted typhus during his research, and he died at the age of 39.
Man, 39.
Yeah. He paid the ultimate price for his dedication to public health. And unfortunately, his death essentially stalled the momentum back in Montana for a long time.
So after 1910, the settlers are still out there on the west side of the valley. The ticks are still biting. People are still bleeding out internally and dying these horrible deaths. The government knows what the disease is, but the science has just hit a wall.
A complete wall.
And bureaucratic inertia sets in. It stays that way for over a decade. What actually breaks the logjam? Because according to our sources, the catalyst wasn't some new scientific paper being published. It was a tragedy.
Yeah. If we look at the history of public health administration, it's a cynical but very consistent truth. Abstract statistics rarely move the levers of massive federal funding.
Sad but true.
An 80% mortality rate among rural homesteaders is a tragedy, but to Washington, it's a localized tragedy. What catalyzes immediate sweeping action is when the abstract becomes deeply personal to the people holding the purse strings.
Right. And in 1922, the disease hits the halls of power. It strikes the family of the Montana governor.
Yeah.
His daughter and his son-in-law both contract Rocky Mountain spotted fever, and the disease, of course, does not care at all that they live in the governor's mansion. They both die.
It is a devastating personal loss for the governor, and the political ripple effect is instantaneous. The state legislature and the federal government suddenly find the political will and the funding that had been completely lacking for a decade.
Funny how that works.
It is. The mandate shifts overnight from, we should study this when we have the resources, to, we need a dedicated team in the valley right now, permanently, until this is solved.
So the PHS is officially mobilized. They decide they need a laboratory right in the hot zone. But I think when we hear the phrase government laboratory today, our modern brains automatically picture like stainless steel, HEPA filters, positive pressure airlocks, people walking around in full Tyvek suits.
Oh, definitely.
But this is 1922 in rural Montana. What does this new federal laboratory actually look like?
It looks like an abandoned two-story wooden schoolhouse.
A schoolhouse.
A literal schoolhouse. They set up shop in Hamilton, Montana, right in the heart of the Bitterroot Valley. They didn't have a time or the budget to build a pristine medical facility. They just needed a roof, some benches, and proximity to the ticks.
Wow.
So they took this old schoolhouse, they moved in their microscopes, their cages of guinea pigs, their glassware, and they just went to work. And it's really difficult to overstate how dangerous that environment was.
I can't even imagine.
They are working with a pathogen that has an astronomical mortality rate, and they are doing it in a dusty wooden building with drafty windows.
So the PHS sends in a bacteriologist named Roscoe Spencer. But the brilliant administrative move here, the thing that I think really cracked the case wide open, is who Spencer ends up teaming up with. He doesn't just work with other doctors from Washington. He partners with a local Montana entomologist named Ralph Parker.
The inclusion of Parker is critical. It really highlights a sophisticated understanding of vector-borne diseases. If you are dealing with a pathogen that only moves through an insect, a bacteriologist alone is really only fighting half the battle.
Right. It's like trying to disarm a biological weapon. Spencer is the explosives expert. He understands the Rickettsia, the payload. But Parker is the ballistics expert. He understands the delivery system, the tick.
That's a great analogy.
If you don't understand exactly how the delivery system functions, you can't stop the payload from hitting the target.
Exactly. And the life cycle of Dermacentor andersoni is highly complex. It actually takes 2 full years for this tick to mature from egg to adult.
2 years?
Yes. It goes through 4 distinct stages.
Oh.
Egg, larva, nymph, and adult. And it requires a blood meal at every single active stage in order to molt and grow to the next phase.
So it has to bite something 3 different times.
Right. And Parker had to map all of this out in the field. He had to understand which animals the ticks preferred at which stage of their lives. And he discovered something terrifying. The Rickettsia bacterium isn't just picked up when a tick bites an infected animal. It's also passed transovarially.
Wait, what does that mean, transovarially?
It means an infected female tick passes the bacteria directly into her ovaries, into her thousands of eggs. So the baby ticks are born lethal. They're infected from the moment they hatch.
Oh my God. So they don't even need to bite an infected squirrel first. They are just born ready to kill you.
Exactly. They are inherently dangerous.
So Parker is out in these steep canyons doing this grueling field work, dragging flannel cloths over the brush to collect thousands of these ticks and bringing them back to the schoolhouse.
Yep.
And Spencer is inside the schoolhouse trying to figure out how to turn these lethal bugs into a vaccine, which brings us to the actual biological challenge of the 1920s. If I want to make a vaccine today, I can use recombinant DNA technology, or mRNA, like we saw recently with COVID.
Right, we have so many tools now.
But Spencer doesn't have any of that.
He doesn't even have a way to grow the bacteria in a dish, as we discussed. It just dies. He realizes that the only place the Rickettsia multiplies in sufficient quantities to actually make a vaccine is inside the tick itself.
Inside the bug.
Inside the bug, which leads to a gruesome, highly dangerous, but absolutely necessary scientific method.
The tick mash.
The tick mash, yes.
Okay, listener, brace yourself for this part.
Yeah.
They spent the tick seasons of the early 1920s essentially running a lethal agricultural operation inside that wooden schoolhouse. They would take live, infected adult wood ticks that were absolutely teeming with Rickettsia, and physically grind them up in a mortar and pestle.
I'm trying to picture the sheer anxiety of doing this.
Yeah.
You have a little porcelain bowl, and you are manually crushing hundreds of tiny hard-shelled insects. Every single time that pestle comes down, you risk an aerosolized droplet of tick juice splashing up into your eye. You risk inhaling it.
The occupational hazard was just astronomical. One slip of the hand, one splash, and you have an 80% chance of dying.
Yeah.
But grinding them up was really only step one.
Right.
Because once you have this paste, this highly infected tick tissue, you have a massive problem. You cannot simply inject this into a human being.
No, because you'd just be giving them a massive, concentrated, lethal dose of Rocky Mountain spotted fever. You're injecting the live killer directly into their bloodstream. So you have to find a way to make the tick mash safe. You have to kill the Rickettsia without destroying the vaccine potential. And the chemical they used to do this was phenol, also known as carbolic acid.
Let's dig into the biochemistry of this because it's really a foundational concept in the history of immunology. Phenol is a harsh, aromatic, organic compound. It's essentially an antiseptic. When you expose bacteria to phenol, it denatures their proteins and it disrupts their cellular membranes.
Okay.
It effectively kills the organism, rendering it completely unable to reproduce or cause the actual disease.
But hold on, if phenol denatures proteins, wouldn't it destroy the very antigens the immune system needs to recognize? Like, if you just turn the bacteria into useless melted chemical sludge, how does the immune system learn anything from it?
And that is the exact tightrope they had to walk. It was an incredibly delicate balancing act of concentration and timing. They needed a phenol concentration that was just strong enough to permanently inactivate the Rickettsia, to permanently stop its internal replication machinery, but mild enough that it didn't completely obliterate the structural integrity of the bacterium's outer surface.
I see.
Yeah.
Because the immune system doesn't actually need the bacteria to be alive to learn how to fight it. It just needs to see the shape of the enemy.
Precisely. The immune system's B cells and T cells recognize specific molecular structures on the surface of a pathogen. We call those epitopes. As long as those surface structures, those epitopes remain physically intact, the immune system can register them, manufacture highly specific antibodies against them, and commit that specific shape to its long-term immunological memory.
It's essentially like creating a mugshot.
A mugshot, yeah.
If the live Rickettsia is a dangerous armed criminal, you obviously can't just throw the live criminal into the police academy to train the cadets. That would be a disaster.
Right.
But if you use phenol, it's like taking a highly detailed photograph of the criminal's face, neutralizing the actual physical threat, and handing that photograph to the immune system. The immune system studies the mugshot, it learns exactly what the criminal looks like, and it prepares its defenses.
That is an excellent way to conceptualize it.
So when the real live Rickettsia enters the body later through a tick bite, the immune system says, oh, I know that face. We have the weapons ready.
That's it exactly. The phenol creates a chemically stabilized, inactivated mugshot of the pathogen. But getting there was incredibly hard. Spencer and Parker spent years trying to get that phenol to tick mash ratio exactly right.
Trial and error.
Endless trial and error. They tested countless batches on their animal models, dialing in the chemistry, trying to find the exact formulation that consistently protected the guinea pigs without accidentally giving them the disease.
And finally, they get a batch they believe in. The animal trials start showing real promise. The phenol tick mash seems to act as a genuine preventive measure. But here is the massive wall you eventually hit in early medical research.
Yep. You can cure guinea pigs all day long, but eventually, if you actually want to save the settlers in the Bitterroot Valley, you have to inject this experimental bug paste into a human being.
And this brings us to a critical inflection point regarding the ethics and the culture of science in the early 20th century. Because today, the pathway from animal trials to human application is, thankfully, heavily regulated.
Oh, yeah. It takes years.
We have multiple phases of clinical trials. We have institutional review boards, double-blind studies. We require informed consent and vast amounts of safety data. But none of that infrastructure existed in the 1920s.
It relied almost entirely on the personal conviction of the researcher. Which brings us to May 19th, 1924.
Roscoe Spencer, sitting inside that dusty schoolhouse, takes a syringe. He fills it with a substantial dose of their experimental phenol-treated tick mash, and he injects it directly into his own arm.
I just want the listener to pause for a second and imagine being in that room. Put yourself in Spencer's shoes. Would you push that plunger?
It's a terrifying thought. You are holding a syringe filled with ground-up bugs that naturally carry a disease with an 80% kill rate. You soaked it in acid, and you think you've killed the pathogen based on some guinea pig data, but you also know exactly what this disease does to the human vascular system.
He had seen it firsthand.
Right. You know the agony of the internal bleeding, the hypovolemia. You know that Ricketts himself died hunting this family of bacteria. And you push the plunger anyway.
It is an astonishing act of bravery.
Yeah.
But we really must understand it within the context of his era. To modern sensibilities, self-experimentation of this magnitude sounds almost reckless.
Totally.
But within the dedicated scientific community of that time, this was often viewed as the ultimate taking of moral responsibility. You did not ask a desperate local farmer to take a risk with a new, potentially lethal substance that you were not willing to take yourself. If you believed your science was sound, you proved it with your own biology. It was a profound demonstration of accountability.
Put your money where your mouth is, basically. So he takes the shot, and then the waiting. He has to just wait to see if the phenol actually worked, or if he just gave himself a fatal case of the fever.
And he survived. The vaccine did not make him sick, which definitively proved the phenol had effectively inactivated the Rickettsia.
Thank God.
Furthermore, subsequent testing and the gradual rollout proved that the vaccine achieved its primary goal. It provided the immune system with that vital mugshot. Now, it is important to be historically precise here. The Spencer-Parker vaccine was not a magic bullet that completely eradicated the infection in 100% of cases.
It wasn't a perfect cure.
No. but it acted as a robust preventative shield. It drastically reduced the severity of the illness if someone was bitten, and it caused the mortality rate to completely plummet. It took a near-certain death sentence and turned it into a manageable medical risk.
Spencer's survival and the success of the vaccine was a monumental triumph. It proved that their years of grueling, terrifying work in that schoolhouse had actually produced a functional weapon against the valley's invisible killer.
It was a massive win for public health.
It was, but we cannot look at this story as just a straightforward, triumphant march of progress. There is a deeply sobering reality to what happened in Hamilton.
There is. The cost of producing this vaccine was devastating. We talked about the lack of modern biosafety containment in that schoolhouse. They were dealing with a pathogen that modern science literally classifies alongside the most dangerous agents on Earth, and they were doing it on wooden desks.
While they were mass-producing this vaccine because they had to physically grind tens of thousands of infected ticks to meet the regional demand, the worst-case scenarios became reality. Several lab workers contracted Rocky Mountain spotted fever in the course of their duties, and they died.
The historical records explicitly honor the specific men who lost their lives in this endeavor: Gettinger, Cowan, and Curley. And it is vital to recognize who these men were. They were not abstract casualties on a spreadsheet in Washington.
No.
They were local Montana residents. They were members of the Bitterroot community who had intentionally taken jobs in that schoolhouse laboratory, specifically to help produce the shield that would save their neighbors.
I think about the emotional toll that must have taken on the community and on Spencer and Parker themselves. Imagine being a family living on the west side of the valley just a few years later. You are taking your child to the local clinic to get this new preventive shot. You know that this vial of liquid might be the only thing standing between your kid and a horrific death.
Right.
But you also know the family of Mr. Curley. You know that the man who physically ground the ticks to make that specific batch died doing it. That vaccine carries an incredible blood price.
It strips away the sterile, sanitized narrative we so often apply to medical history. We like to imagine scientific breakthroughs as these bloodless eureka moments happening in pristine, glowing environments.
Like in a movie montage.
Exactly. But the reality, especially in the early days of public health, was often grueling manual labor that demanded profound sacrifice. These men were essentially working in the trenches of a biological war. They knew the risks, they saw what the disease did every single day, but they kept grinding the ticks. They gave their lives to build the infrastructure of survival for their entire region.
And that infrastructure did survive, and it scaled up remarkably. Let's look at the legacy of what they built. By 1925, just a year after Spencer's self-experiment, the PHS began officially vaccinating residents in the Bitterroot Valley. By 1927, production had ramped up enough that the Spencer-Parker vaccine was broadly available to the public.
The scale of the rollout was just a massive logistical achievement. By 1940, approximately 500,000 people in the Rocky Mountain region had received the vaccine.
Wow.
500,000 people were given that immunological mugshot.
It fundamentally altered the trajectory of the American West. The west side of the Bitterroot Valley was no longer a quarantined graveyard. Families could farm, children could play in the canyons, the regional economy could function without the constant hanging dread of an 80% mortality rate. And as the vaccine proved its worth, the federal government finally acknowledged the reality of the situation in Hamilton.
They realized that the scientific operation Spencer, Parker, and their fallen colleagues had built was far too valuable and far too dangerous to remain in an old wooden schoolhouse.
So in 1928, the government finally allocates the funds for a permanent upgrade. A purpose-built, modern brick laboratory replaces the schoolhouse. It officially becomes the Rocky Mountain Laboratory. The U.S. Public Health Service had successfully established a permanent, state-of-the-art research outpost deep in the western frontier. But here is where our sources point out a fascinating twist in the institutional history.
And this is the crux of the overarching historical narrative here. The Rocky Mountain Laboratory was conceived, fought for, and built with the blood and sweat of the Public Health Service. But institutional bureaucracies are dynamic, shifting entities. In 1937, less than a decade after the new brick building opened its doors, a major reorganization occurred within the federal government.
Okay, what happened?
The Rocky Mountain Laboratory was formally absorbed into the National Institutes of Health, the NIH.
The NIH, which at the time was growing, but eventually becomes the absolute behemoth of global medical research that we know today.
Exactly. Under the umbrella of the NIH, the Rocky Mountain Laboratory's mandate expanded exponentially. When World War II broke out, that facility in Montana pivoted. It utilized the exact same mass production techniques they had developed for the tick mash, but adapted them to become a massive vaccine factory for the
Well,
military.
he didn't know that.
Yeah, they churned out millions of doses of immunizations for typhus and yellow fever to protect Allied troops deployed across the globe. Today, the Rocky Mountain Laboratory is one of the premier high-containment BSL-4 facilities for the NIH's National Institute of Allergy and Infectious Diseases. It is where scientists go to study Ebola, Nipah virus, and SARS-CoV-2. It is an absolute crown jewel of modern biomedical infrastructure.
Which brings us to the central irony of this entire deep dive. We started by noting that historically, the PHS held the regulatory authority they quarantined, they observed, while the actual hands-on bench science happened elsewhere.
But Rocky Mountain spotted fever is the grand, towering exception to that rule. In the Bitterroot Valley, the PHS officers were the ones who rolled up their sleeves. They were the ones who self-experimented. Their local technicians were the ones who died. The PHS built a functional, world-saving vaccine from scratch in the most hostile conditions imaginable, generating an immense, hard-won scientific capacity.
But true to the historical patterns of government bureaucracy, the agency that does the frontier groundwork rarely gets to keep the sprawling estate that grows from it.
That's usually how it goes.
The heroism, the grit, and the ground-level sacrifice belonged entirely to the public health service. But the lasting institutional inheritance, the pristine modern laboratories, the massive federal budgets, the global prestige was ultimately absorbed by their offspring agency, the NIH. The NIH is the institution that inherited the house that Spencer and Parker built.
It is a profound legacy. The modern NIH stands quite literally on the foundations laid by Roscoe Spencer, Ralph Parker, Gettinger, Cowan, and Curley.
We have covered a massive amount of ground today. We started by looking at a terrifying geographic puzzle, a valley where living on the west side of a river meant a near-certain death from internal bleeding and organ failure. We traced the brilliant but tragically interrupted work of Howard Taylor Ricketts, who identified the tick vector and the microscopic pathogen, only to lose his life to a related disease down in Mexico.
Such a loss.
We saw how a tragedy in the governor's family finally catalyzed federal action, leading to the brilliant pairing of a bacteriologist and an entomologist in an abandoned schoolhouse. We broke down the intense biochemical challenge of using phenol to denature the lethal Rickettsia just enough to provide the immune system with a molecular mugshot without destroying the evidence.
The science was just incredibly elegant for its time.
It really was. We witnessed the quiet, immense bravery of Roscoe Spencer risking his own life in 1924 to prove the science actually worked. We honored the devastating sacrifice of the local lab workers who died producing that preventive shield. And finally, we watched that wooden schoolhouse evolve layer by layer into a modern, high-containment crown jewel of the NIH.
It is a testament to what human beings are capable of when pushed to the absolute edge by their environment.
It is, and I want to leave you, the listener, with a final thought to mull over. When you see news reports today featuring massive, modern institutions like the NIH or the CDC, when you see those pristine stainless steel high-containment labs, the multi-billion-dollar budgets, the scientists working in pressurized hazmat suits, it is incredibly easy to view them as detached, sterile bureaucracies. They feel completely removed from the messiness of the natural world.
They feel very clinical.
But after exploring the Bitterroot Valley, you have to ask yourself, how does it change your understanding of modern medicine to know that the foundations of those towering, pristine institutions are not made of bureaucratic paperwork? They are literally built out of the dust of abandoned wooden schoolhouses. They are built on the terrifying personal risks taken by desperate scientists. And most importantly, they rest entirely on the quiet, lethal sacrifice of local workers who were simply trying to stop the bleeding of their neighbors.
It really changes the perspective.
Every vial of medicine, every vaccine, every preventative measure we take for granted today has a lineage. And sometimes if you trace that lineage back far enough, you don't find a corporate boardroom. You find a guy in a drafty room in Montana, grinding up bugs in a bowl, betting his life that he can save yours. Thank you for joining us on this deep dive into the archives. Keep questioning the history behind the science that keeps you safe, and we will see you next time.